Welcome everybody. This is another lecture of Environmental Soil Microbiology, SWAE3411. Today's lecture will be about soil as a microbial habitat. What is it with soil that allows it to harbor such a big biodiversity of microbes, and how the microbes are conditioning the soil and making the soil their habitat. You are making up, for example, making up the structure in the pores of the soil, and also decomposing organic matter, producing humus, and among other ways. Among other ways we're going to talk about now. But we're going to talk about how microbes are conditioning the soil, and also how the soil conditions are allowing some types of microbes or not to live on these environments. So what are these factors? We can classify these type of factors in intrinsic factors and extrinsic factors. So the intrinsic factors will be the factors related to the properties of the microbes themselves. And so, for example, the ability to make spores, for example, from fungi, or endospores from bacillus and actinomycetes, will allow these microbes to live in more extreme conditions. For example, if you have drought cycles or more heat on the environment. So the size of the microbes also will tell where these microbes, what type of pores and conditions these microbes can occupy, the motility, if they can move around in the soil, and the type of motility will be very important. Structural features from these microbes, biochemical capacities are very important. For example, if they can use different molecules, organic or inorganic, as a source of energy, what are the oxygen requirements that these microbes have. So these are very important features, intrinsic factors. And we have also that some microbes, they will form associations among themselves and among them in mesofauna and plants, and will allow these microbes to occupy some regions of the soil that otherwise they wouldn't. So about extrinsic factors, some things on the soil are very important for the microbes being able to occupy and survive this environment. For example, organic matter, soil structure, soil atmosphere, soil water, the moisture regimes that you have on the soils. The pH of the soil, very important, the temperature, oxidation reduction potentials, radiation from if it's in the soil surface, wind and relative humidity also will play a role. So when we talk about soils, you will think about chemical characteristics, and soil abiotic environmental factors, that will be conditioning the soil biota. So there's a complex interchange between the microbes and the soil abiotic conditions that you have there. So when just a quick outlook and revision, when we think about soils, you have to think about the pores in the solid phase. The soil pores will be occupied by air and water in different proportions depending on the moisture content. And the solid phase of the soil will be composed of organic matter and mineral particles. Organic matter will be the fraction that will be conditioning soil microbes in more strongly, more than the mineral particles. But also mineral particles will condition soil microbes. The texture of the soil will be important for the type of microbes and the diversity of microbes, but not as much as the organic matter content of these soils. So when we talk about organic material, in general, we are talking about all organic material, but and the biomass, the living organisms, which are a part of the organic matter of the soil, this biomass, they will be interacting with the other part of the organic matter, which are fresh organic matter. The fresh organic matter are plant and animal residues, fresh residues that have not been decomposed fully. And the humus. The humus is the end product of the decomposition of the fresh organic matter. So this humus, they are less susceptible to be decomposed by the microbes, and they condition the environment for the microbes or for the plants. Yeah? So what is humus? Humus is what's left after the biological breakdown of plant and animal material, fresh materials. And what makes this breakdown? Of course, soil heterotrophs. Yeah? The heterotrophs in the soil are the microbes that are using the organic matter as a source of carbon. Most of the heterotrophs, they are chemoorganotrophs, so they use the microbe, the organic matter, the fresh organic matter, also as a source of energy. And as they do that, they will leave the humus as an end product of this degradation. They will not be able to eat all the organic matter from the soil, and therefore what is left is a complex, very resistant type of organic matter, which is variable on its chemical properties and chemical structures. And this organic matter is what we call humus. How do microbes form humus? The answer is by releasing enzymes. And just a reminder of what enzymes are. Enzymes are proteins. They're chemically active proteins that are able to catalyze chemical reactions. So they accelerate these chemical reactions. The enzymes have an active site, so a place on the surface of the enzyme which attracts the substrate, binds to the substrate, and is able to transform chemically the substrate into the product of this reaction. So microbes, they do this in order to break down the fresh organic matter and release the carbon and the nutrients they need to survive. Yeah? The carbon and the nutrients they need to survive. So examples of enzymes, you have, for example, cellulase, which breaks cellulose, urease, which breaks down urea, and phosphatases, which releases the phosphorus bound to the organic matter, the phosphate groups bound to the organic matter. But these are just examples. There are thousands of enzymes on the soil that have microbial origin, and they're very important for the biochemical properties and capacities of these soils, or the capacity of the soil microbes to digest the organic matter and make these nutrients back available to the microbes and the plants. So why we care about this? First of all, the humus or humic substances, they are quite relevant to the quality of the soils. Yeah? So they are very resistant to decomposition. They form complex with silicate clays, and they have a lot of charge. They are colloids which have a lot of pH-dependent charge. And the amount of humus we have on the soil is perhaps the single most important measure of soil quality that we have. The soil health and soil quality can be directly correlated with the amount of humus you have on the soil. So the more humus you have, the better the environment will be for microbes and plants. In Oman, of course, you don't have a lot of humus, you don't have a lot of organic matter due to two factors. One, you have very sandy soils, and the other one, the climate is arid. You know, not a lot of biomass growing on the surface of the soil to be cycled and build up this organic matter, and the conditions of temperature are not very conducive of the accumulation of this organic matter in the soils. But this organic matter is a source of energy for microbes. It provides nutrients for these microbes and for the plants. It allows the soils to build up structure, absorb pollutants, pesticides, and all this cation exchange in the soil is what makes the soil so useful as a nutrient reservoir for plants. So the organic matter is building up structure, but by binding to the other colloids for the clays in the soils. But organic matter in itself in the clays, it's not only a chemical process. The chemical process which makes up the structure in the soil is what we call flocculation. But this flocculation of colloids, these colloids binding to each other, it's not the only process that is happening. The aggregation of the soil is highly dependent on the soil biota, and that includes the plant roots and microbes like fungi, bacteria, and among bacteria, actinomycetes. So the formation of soil structure is influenced heavily by soil microbes. The growth and movement of these organisms in the soil, including the roots and microbes, it's very important to shaping the type of pores that you have on the soil. There's a process that we spoke before. This process of bioturbation is very important because it's the ability of this meso and macrofauna to mix the soil with the organic matter from the surface, allowing this organic matter to reach down in the soil and form this structure. So the bioturbation, the mixing of the soil with the organic matter, is very important for the formation of soil structure. And this is basically a process which is regulated by macro and mesofauna, earthworms, for example, or in other mesofauna and macrofauna like ants, termites, Soil organisms, they will break these residues, and they will, the microbes, the bacteria in specific, they will produce extracellular compounds, glues, that stabilize these aggregates. The fungi will produce even proteins like glomalin that will stabilize aggregates. Additionally to these extracellular carbon-based molecules that are helping to stabilize the soil aggregates, you have the fungi and actinomycetes will provide physical structure stabilization to these aggregates by the hyphae that is surrounding these aggregates, make this package and holds these microaggregates together to give them more stability. Yeah? So in specific, we have that fungal hyphae, actinomycetes, filaments, bacterial glues, and the organic matter together will be responsible for the stabilization, the formation and stabilization of these aggregates in soils. And these aggregates will be responsible for creating the aeration conditions that you have for these microbes. So here's some microscopy of these actinomycetes, filaments, and hyphae in bacteria glues that are being deposited on the surface that are being responsible for the aggregate formation in soils. So about the size of the aggregates, we have macro aggregates, which is around the size of 3 millimeters. Among the size of 0.3 millimeters, we have micro aggregates, and 0.03 millimeters will reach the size of submicro aggregate. And then below that, you will think about primary particles by themselves, not about the aggregates, the micro aggregates. And one of the important things about these aggregates is the formation of these aggregates condition the size of the pores we have in the soil. So if we have more macro aggregates, we have more macropores. And if you have less aggregation, you will have more micropores. So the size of the pores are heavily conditioned by the types and the amount of aggregation that you have on the soil. And this aggregation of the soil, it's heavily conditioned by the microbial activity and by organic matter especially humus, which is a product of microbial degradation of the fresh organic matter also. So this is all conditioned by soil microbes and also by the presence of other colloids in the soils. So the size of these aggregates will condition what type of microbes live there. We have in this photograph here this small in this circle you have on the electron microscopy here on the right, there is one bacterium, and it's on the side of a quartz silt particle. So you see how these micropores could be a microenvironment for colonies of this bacteria, for example. Whereas if you have macropores, this will be conditioning the life of other types of microbes. So the size of the pores condition the airflow, the moisture concentration, the water availability to these microbes, and how much does it oscillate these conditions? Does it have a stronger drought cycles, or just it keeps moisture for a longer period? So the size of these aggregates regulates what are the conditions for the microbes to be able to survive. So different types of microbes will be able to live in different size of aggregates. And here is also showing what are the size of the pores, and relating that to the size of, for example, bacteria, which is roughly between 1 and 5 micrometers. Yeah, so they're the size of medium pores. Yeah, they're on the range of medium pores. And very fine pores, only the very small bacteria will be able to colonize these very fine pores. So the other property of the soil that is conditioning the growth of microbes is the soil air. Yeah, the soil air. And the composition of soil air is very different from the composition of atmospheric air. The oxygen on the atmosphere is about 20-21%. In the soil is always lower than that, and it becomes lower and lower as you go deep. Carbon dioxide is 0.04% on the atmosphere, and you can have a build-up for up to 10% of carbon dioxide in the soil. So the microaerophilic microbes and the capnophilic microbes can find their environments on these soils. Anoxic microbes will find their sites on the soils because of the low oxygen concentration in some areas of the soil, some pores, depending on the moisture condition and the airflow. And also the facultative anaerobes also will be able to colonize. There are other type of gases that you find on the soil atmosphere, the soil air. For example, methane can be formed by anaerobic decomposition of organic matter by archaea, and hydrogen sulfide also will be formed by the reduction of sulfate on these soil environments. Hydrogen sulfide will be toxic to several microbes, and methane will be also problematic. Some microbes can need the methane to live, but others would not like the environment with high methane. N2 gas is fairly similar to the atmosphere, and it's fairly inert, so there's not much to say about this. Just that you will have nitrogen fixing bacteria that will use this nitrogen gas, but the concentrations of N2 will not change too much, and it will not alter microbial populations on the soil. So talking about the air, we think about from the surface of the soil, the oxygen is diffusing in the soil, and carbon dioxide is diffusing out. Also, methane will be diffusing out, and hydrogen sulfide will be diffusing out, depending on how waterlogged the soil is. And because of that, you will see that the concentrations of carbon dioxide, they will vary from the surface to deep on the soil. And the deeper you go, the higher you have the concentration of carbon dioxide. So this gradient of carbon dioxide concentration will be conditioning also the type of oxygen requirement that these microbes have on the soils. And close to the surface, you have more aerobic conditions, and as we spoke before, the aerobic microbes that need oxy conditions, that need oxygen for oxygen for the respiration, they will be more fast growing than the anaerobic microbes. Yeah. Clay particles will influence, but will not be the main characteristic of the soil affecting soil microbes. They will condition the soil microbes by being a reservoir of nutrients, by creating those microcytes, micropores for some special types of bacteria. If you have a lot of clay on the soil, also you will have more anaerobic conditions, and you will create those microcytes for anaerobes. And also there is all this ability of absorbing chemical substances, but also not only chemical substances. Some clays, some microbes will have strong interaction with clay surfaces and will coat themselves and will bind and absorb the full cell into clay particles. And if it's a nanoclay, the nanoclay will be coating the microbes. If it's a quite large clay particle, the microbe can bind it in a similar size. But the coating of microbes with these clays could help the microbes to stabilize themselves and protect them against the predation from other microbes. All right, so the other factor from the soil is affecting microbial growth is temperature. In general, you will have that near the surface, you will have the soils will be hotter, and then it will allow the microbes to grow faster unless it's too hot. If the soil heats too much on the surface, like in desert soils of Oman, you will have the presence of thermophiles. The thermophiles will be colonizing the topsoil because of the strong heating of the soil surface. But if you think about the temperature in around the world, normally soil will the hotter the soil is, the better for the microbes. And the microbial growth will be benefited by this higher temperature, and also all the biochemical processes from these microbes will be benefited for having a higher temperature in the soils. So because of that, you have near the surface of the soil, you will have that you have more oxygen, more organic matter. You will have higher temperatures, and due to that, you will have a higher microbial biomass. But not only a higher microbial biomass, you will have also a higher microbial diversity, and a higher diversity of functions, of microbial functions near the surface of the soil. And this is one of the reasons that is so important for us to conserve and prevent the soil from being eroded, because this topsoil, it's not only retaining nutrients, but it's retaining all the biochemistry, the microbial diversity, and the microbial population near the surface of the soil. So we need to think about the function of the soil as the ability of the soil of providing those nutrients, providing those conditions to the plant, cycling that organic matter. If we erode that topsoil, we're losing soil function at the same time. So just a clarification topic, because this is discussed in other lectures. Biomass is proportional to the number of cells. We're talking about the amount of microbe mass that you have on the soil. But when we talk about diversity, we are talking about number of taxa, number of taxa of microbes. How many different species do we have of microbes in the soil? And when we talk about functional diversity, we're talking about how many different processes the microbes can carry out on this soil, how many different processes. And there's another concept that we need to come back and highlight again, is the concept of functional redundancy. If we have a high number of taxa, if we have a high diversity, then it is more likely that we have many microbes doing the same function on the soil. If we have many microbes doing the same function on the soil, then we can afford to lose some of these microbes to some extreme events like drought, or high temperatures, or flooding, for example. Because if we have a lot of redundancy on the system, we will still keep the function if we lose some of these microbes. But if we have that some of these microbes, some of the functions are carried out by few species of microbes, we are in a higher risk of losing that function when we have extreme events on the soil. And normally when we talk about microbial diversity and functional diversity, we use to discuss these diversity indexes. And the diversity indexes will not only be a function of the number of taxa, but also the proportion or the dominance of this taxa on the population. Because we may have a high number of taxa, but one taxa occupies half of the population of the biomass of the soil. If we have that one taxa is very dominant, then also we will say that the diversity of microbes is low. So it's the diversity indexes, they integrate the number of taxa, but also the dominance of this taxa. If we have the biomass is divided more evenly between all the taxa, and we have higher diversity if we have those conditions. The next thing on the soil which conditions the growth of microbes is plant roots. So plant roots are very important for soil microbes. In the soil that is surrounding the roots, that's the region of the soil that we call the rhizosphere, that 1 or 2 millimeters of soil that are directly on the side of the roots, they are completely different as an environment for the rest of the soil. So we tend to separate this zone as rhizosphere and the rest of the soil as bulk soil. And then we can see how important this rhizosphere is, because the plants are feeding the microbes, exudating carbon-based molecules like organic acids, in the soil. Also, for example, surfactants or siderophores are being exudated into the soils, and the microbes will be interacting with these root exudates, feeding on those root exudates, and can have a very strong interaction with the roots, benefiting the roots on these systems. So some of the things that microbes can do for the roots on the rhizosphere is protect the roots from pathogens, from other microbes there, or that could be damaging the plants. Also, mobilization of nutrients could be an important factor. So if the microbes are benefiting from the roots, they want the plant to put more roots on the soil. So there is this protocooperation happening on the soil, where both the microbes and the plants can live by themselves, but if they cooperate, then they are better together. They're better together. And of course, we have to mention here, as we spoke before in the class of soil fungi, the mycorrhizosphere. So some of the roots, the majority of the roots from higher plants, they are associated with symbionts, fungi symbionts, and this fungi that are associated with roots, they are called mycorrhiza. And this mycorrhiza will extend the ability of these roots for exploring the soil volume and exudating materials in the soil. So you have the rhizosphere, but you have also mycorrhizosphere, which are the region associated with the mycorrhiza, which are by themselves associated with the roots. Yeah. So here we are looking at the biomass of different microbes of the microbial biomass associated with different plants and in the book soil. And then we will see that the biomass can be more than 10 times higher on the rhizosphere than it is on book soil, depending on the plant and how active the root exudation is in these different plants. Next thing we do have to think about in soil is the water on the soil and the ability of the microbes to have access to this water. And the science of this is similar to the science of what is the water availability to plants also. It's not, when we think about the water availability, we're not thinking about the moisture content on the soil by itself, but we're thinking about the water potential in soil. Especially, two potentials are very relevant on these systems for the microbes, which are the matrix potential and the osmotic potential. Yeah. The matrix potential is the attraction of this water for the soil surfaces. It's the sponge effect on the soil. And the osmotic potential will be a function of the amount of solutes that you have on this soil water, which will prevent the microbes from absorbing this water because they need to filter out the salts on the soil solution. And then will create this difference of potential due to different concentrations of salt or different ionic strengths inside the cell and outside the cell. So if you have saturated conditions, you of course have low oxygen concentration. And then if you have very dry soils, you will have low water availability for the microbes. So you move from an extreme condition where you have very easy water availability, but low oxygen availability to microbes. In the other extreme, you will have low water availability, but a good oxygen availability for microbes. So in the two extremes, you will have this variation of not only water availability, but also oxygen availability. And this relationship between the moisture in the soil and the potential, it's what we call the matrix, the soil water characteristic curve, which is a property of each individual soil, and it's related to the texture and the amount of organic matter that the soil have. The size of the pores, the distribution of the size of the pores will be very relevant to the shape of this water, the potential and the moisture content relationship in the soil. And we have that normally marked that at the minus 10 kilopascal will be the field capacity. It's about what the soil can retain when all the gravitational water is drained. And when the soil is very dry, that will be about 1,500 kilopascal. So that will be very dry, and the water will be not available for plants and also not for microbes. Yeah. So the microbes, they have a relationship with the water potential. If the microbe can withstand very negative soil water potentials, these microbes is what we call xerophiles. Yeah? And this negative water potential, if it's caused by high salinity, not by low moisture concentration, this type of microbes are what we call halophiles. So if it is a very negative osmotic potential, then we call the microbes halophiles that can withstand these conditions. And if it's a very negative matrix potential, then we are talking about xerophiles. So of course you will make the link that many of the halophiles are also xerophiles. The only difference will be that some of the halophiles, they will need to cope not only with the low water availability, but also with ion toxicity from the salinity. And this high concentration of ions will have toxicity for many microbes, but also will inhibit some of their enzymes and will generate more energy demand for these microbes to pump out the salts and be able to cope with the high salinity. So of course, if the cells dry to a certain point, we have what we call plasmolysis, which consists of the loss of water from the cell to the environment due to the very dry, very low potentials outside the cells. We can classify also the microbes for the range of, on the wet range of these soils. If the microbes, they like very wet conditions, then we call these microbes hygrophiles. In intermediary conditions, it will be mesophiles, and as we spoke before, xerophiles will be the ones withstanding high drought. And the bacillus and actinomycetes will be typical xerophiles on soils, and also many fungi will be able to withstand these conditions. Now, to finish, let's talk about what are the factors that are most influential on the microbial diversity. Not biomass, but diversity. And the diversity of archaea, fungi, and bacteria will be very differently regulated by different soil factors. If we talk about the whole diversity of the soil, including bacteria, archaea, and fungi, normally, in the majority of the studies, we will think that total organic carbon will be the main factor regulating the diversity of these microbes. Because the majority of this biomass, the bulk of this biomass, they're heterotrophs. They're using the organic matter as a source of carbon. So the more organic carbon that you will have a higher biomass and a higher diversity of these microbes. But when we speak about bacteria by itself, bacteria is not only regulated by soil carbon, but actually if you look across very extreme environments, very different environments, not only, for example, not in a study not only across a single country, but across different ecosystems and continents, what we will find is that the total organic carbon will not be anymore the thing conditioning the diversity of bacteria, but salinity will be the main factor conditioning the diversity of these bacteria. So for bacteria, salinity is the main factor, but when we talk about microbes in general, it's generally assumed that total organic carbon is the main factor. So more organic matter will increase the diversity of microbes in the soil. More salinity will decrease the diversity of bacteria in the soil, but will in turn increase the diversity of archaea. Okay, but archaea is not. There are not many studies, and the studies using 16S genes, they're normally not very convincing for archaea diversity. But the little information we know is that with high salinity, you increase the diversity of archaea. They tend to like more extreme saline environments. As the proportion of bacteria decreases, the proportion of the diversity occupied by archaea is increasing on the system. All right, this is all I have for you today. Thank you very much.